welcome back. It's a great pleasure to introduce you Professor José Alexandre Diniz-Filho. Professor José Alexandre has a biology degree in the Universidade Federal de Sergipe. It's one of the states in the northeast of Brazil. A master's and a PhD degree in zoology by Universidade Estadual Paulista Júlio de Mesquita. It's Unesp in another campus. And currently he is a professor in ecology and evolution at the Universidade Federal de Goiás, another state in the center of Brazil. José Alexandre works in the interface of ecology and evolution to understand biodiversity patterns to provide information that can add in conservation, such as patterns and process that cause them. His lab uses different approaches ranging from population genetics, community, and geographical ecology, and also phylogenetics and comparative methods. And I love his book, by the way. So it's a great pleasure for us to have you here. Professor José Alexandre, for those who don't know, Professor José Alexandre is one of the most important ecologists in Brazil. So thank you very much for accepting our invitation. And so help me to welcome Professor José Alexandre.
Okay, thank you, Flavia, for the invitation. It's a pleasure to be here. I hope you forgive my English is a bit rusty, I think, after 2.5 years of pandemics, but I trust you can understand me, hope so. Thank you also, Ricardo, for the possibility of talking to you here in this workshop. It's a very nice work that Serrapilheira is doing, very, very, very nice. And when Flavia asked me to talk to you, I was wondering about talking in terms of theory models and methods in macroecology. As Flavia said, in our lab, we work with different methods, different approaches in terms of simulation modeling, statistics applied to solve different problems, but mainly in terms of broad-scale ecology and evolution and the interface. And in the last few years, I'm quite interested also in the more philosophical or epistemological issues underlying macroecology. And I think that we have some very, macroecology provides a very interesting example of how these different approaches, philosophical approaches, can be better understood, and how they, and in particular, simulation models, as we're going to see, provide a very interesting interface between different ways to think about theory and models in ecology and evolution. So, oh, sorry, something happened here. Are you getting the screen or disappeared?
Oh, sorry. Now again, yeah.
Okay. We have a small delay, but no problem. So first of all, what we're talking about when we think about macroecology. The term macroecology appeared in a paper published in Science in 1989 by James Brown and Brian Maurer. And basically the idea is to study broad ecological systems at much broader scale than it was discussed at that time. And of course, during the next years, several, the field started to grow, and we had several textbooks. For example, Brown in 1995, Maurer published his own book in 1999, and we have the British ecologist Gaston and Blackburn mainly published several papers, many papers, and several books from 2000 on. And the overall idea of macroecology is to have to couple the non-experimental and statistical investigation of the broad-scale relationships between different variables that are used as surrogates of population dynamics and interactions. And so we try to merge the standard process that are studied by ecologists at small scales, such as interaction, population dynamics, and population fluctuations, extinction, local extinctions, and so on, with broad-scale processes that are usually studied by biogeographers or paleontologists, macroecologists, in terms of speciation and extinction. So the idea is to use different approaches at very broad scales to understand the ecological process in deep time and in a continental, for example, geographical scale. And basically, the initial idea was to study some patterns, for example, that actually were developed much earlier. For example, species area relationship, species abundance distributions, the relationship, for example, between geographic range size, body size, or abundance and body size across different species. And also to help understanding the latitudinal diversity gradients that was a pattern that was studied since 19th century by biogeographers, but now studying this pattern in a more ecological and processes based basis, right? And more recently, we had a very interesting review by Brian McGill, that is now the editor of the Global Ecology and Biogeography journal. That is the most specific journal in macroecology. And Brian defined here macroecology in a slightly more different way, thinking first in terms of broad-scale patterns, right? So that's the standard view of macroecology, the broad-scale patterns, ecological and evolutionary patterns, but also as macroecology as an emergent non-reductionist approach to study these patterns. So finding fundamental laws and principles that would drive these patterns at broad scale. So we have these two slightly different perspectives to understand macroecology. And so I'm going to start very, very briefly to talk about the epistemological basis that we have been thinking about in terms of what these two definitions of macroecology and how they can be interpreted in a more epistemological basis. And for this, I really like Ronald Geer's idea of what he called scientific perspectivism, right? So macroecology is actually a perspective, a way to view ecological data. And in particular, this kind of view leads to some ideas in terms of constraints of what we can do and what we can't do in terms of hypothesis testing and in terms of establishing causal relationships and what is the underlying principles behind all these macroecological patterns. But basically, what I want to show you and to discuss is that we have, for example, this is the basic framework that Ronald Geer defined. We have a general theory, and then we can go to what he called the representational models, right? So we have very abstract principles in terms of theory, and we go to representation models. And these models allows what we can talk in terms, a very general idea of hypothesis testing. So one of the most interesting things here is that Geer started to think that theory, in a very broad sense, theory is a very complex and a very vague, used in some way ambiguous way in the literature. And we gradually moved to go beyond the overall idea of theory to focus on models. So the idea is that we can go directly from general principles and general concepts, general ideas, to what Geer called representational models. And we also have a very interesting idea here, is that he called that, he told that in general, we do not have really observational or empirical data. What we really have is a model of the data, right? For example, in our case in macroecology, when we are thinking about diversity gradients, and we have, for example, a map just like this that you are seeing here in the screen. This is the diversity pattern of amphibians at global scale. We have more species in the tropical regions. We have more species in the Amazon and in the Atlantic forest. Okay, but this is hardly observed. We do not observe this. What we have are species occurrence, then we have to build the geographical ranges, then we have to overlay these ranges to get a broad scale, a macroecological estimate of number of species diverse or species richness more specifically. So this is not actually empirical in terms of observation. We talk about empirical because we do not have a better word perhaps, but this is actually a model of data. So the idea of hypothesis testing and then generalization to achieve some predictions is to compare representational models with models of data, right? And these models of data represent the real world, whereas the representational models represent the theorizing, the overall ideas, abstract ideas that we have about this, the theory, the explanation, the underlying explanations for the patterns, right? But we can go a bit further and we can say that theories are not necessary, right? So that's why I'm talking about the more general term theorizing process, because actually the models are the more important things. And so this was some kind of revolution in terms of philosophy of science that was mainly triggered by this book by Nancy Cartwright, How the Laws of Physics Lie, that when she proposed that models are the most important things. And also the book by Margaret Morgan and Mary Morgan and Margaret Morrison in 1999, where in this book they proposed that models are what they call it the mediators between theory and reality. And theory is a very vague, very abstract, usually very confusing term. And so models are the most important thing. So models as mediators, because we can even learn about the process, about the patterns in the process, for example, in macroecology, using the models. So we are not focusing on the real world, we are focusing on the models. So models gain much more importance from the 2000s than the overall idea of theory. And I think that this framework is particularly interesting in macroecology. And in an extreme, this we can see all this idea as providing a different way to view the entire scientific activity. So the most standard view of science is what we usually call theory-based science. We have the theory and the models are derived from the theory. But actually what they are proposing, for example, Nancy Cartwright, Margaret Morrison, and all these people, what they are proposing is that the focus is the model. So we have what we usually call model-based science, right? And we have a very, very interesting recent review of all this idea, what sometimes is also called the pragmatic view of theories, in particular in ecology from my friend Paulo Inácio Prado and Charbel and other people. Renata and Bruno is the leading author, and they published this paper last year in Oikos. It's very interesting in terms of discussing these alternative views of how scientific theories can be better understood and how and the whole of models in terms of better understanding patterns. And this is very, very important in macroecology because as we are talking about broad scale evolutionary and ecological patterns, for example, we can't do experimental approaches. We can't do experiments. Only a few processes or a few minor ideas, but we can't do experiments. And we also have a very deep time scales. We are talking about very large plates, very large, very dynamics in deep time. So it's very complicated to establish and to define causal relationships, right? And so let's go back to diversity gradients and see how we can understand diversity gradients in terms of this perspective that Ronald Geer and all this model-based science developed. And as I told you, in our lab, we have been working with diversity gradients for a long time, and we use different approaches to investigate diversity gradients, ranging from very standard statistical analysis, mainly focusing on spatial statistics to understand patterns of diversity, up to simulation models that will be the focus of my talk here today. But as I told you also, diversity gradients were discussed in the ecological and biogeographical literature since the 19th century, for example, with von Humboldt, that proposed that the explanation for diversity gradients is basically temperature, right? Von Humboldt came to South America and was mesmerized by the high diversity in the tropics. All naturalists from the European naturalists from the 18th and 19th centuries were find the biodiversity in the tropics a wonderful thing. And von Humboldt was the first to propose or to talk that the explanation for the extremely high richness in the tropics was temperature. And since 19th century, hundreds of hypotheses were proposed to explain the diversity gradients, but from 2000s on, researchers started to reduce the number of more plausible explanations for this kind of gradients in species richness to a few more likely and a few more testable ideas. For example, habitat heterogeneity and complexity, biotic interactions, the effect of area influenced by MacArthur and Wilson's equilibrium theory in biogeography, the idea of diversification rates, speciation and extinction, the age, tropics tend to be older than the temperate regions, climate, right? So we have these ideas that are rounded by a red circle, which appears at much broader scales, and that we usually think a bit more about the effects of climate. And more recently, when we started to have more phylogenetic information, we have to have more conditions to test the ideas of niche conservatism, which is related to age of the clades, and more recently about diversification rates, right? But even when we talk about these three possibilities that are more likely at broad scales, we have a much more complex interaction between different variables that we can use as surrogates of the different processes, ecological and biogeographical, or ecological and evolutionary process that would underline these gradients. As you can see here, one example of these possible links between, for example, temperature, productivity, area, or climatic stability, with all this process leading to different patterns in geographical patterns in speciation and extinction, and this lead to different gradients in terms of the number of species that we can find across the globe. And so the idea is that we have to start thinking about this, and the simplest way to think about this is to measure this kind of variables and try to do some correlations, right, between these variables, and we can use even more sophisticated methods such as structural equation modeling and this kind of approach if we have this kind of network of relationships between the variables and the response variable that would be species richness. So these correlative approaches were the standard way to investigate diversity gradients using spatial statistics or structural equation modeling for a long time. But there was a claim that we should go to a more theoretical approach in terms of the theory. So we go back to that scheme by Gehr. And one of the main ideas that was a more interesting and a more proud ways to do this was proposed by James Brown, that was the creator of the term macroecology. And Brown developed what he called a metabolic theory of ecology. So it's a very general theory in terms of how energy is used by the organisms. And Brown proposed that these patterns, this process of energy use can explain most, if not all, ecological patterns. So we have a general theory, and derived from this very broad theory in terms of how energy enters in the system, in the ecosystems, and drive ecological patterns. One of his theories, the constitutive theories that they call it, is a theory to explain the diversity gradients, right? So he proposed one of the models that is derived from the theory that Nancy Cartwright calls an interpretative model, as an interpretation of the theory. They derived a model that says that the relationship between the logarithm of species richness against the inverse of temperature has a particular slope. So it's a very interesting model because it's very testable, right? So they develop this model and they say, okay, we now can understand the number of species of ectotherm organisms by using only the temperature, right? So we have this particular slope that was in this first version was equal to -9. So we have an inverse relationship between species richness and temperature, right? So this is a very, it was a very different way to think about the diversity gradients, because they are not proposing actually a correlative model, right? Statistical model. They are proposing a theoretically oriented interpretive model that do a very precise prediction about the diversity gradient. And this, and they proposed this in a paper that was published in Science in 2002. And they did a few tests, and okay, as you can see here, this is a very nice approximation of the slope. The empirical, or the not observed, but the empirical slope was very close to what was predicted by the theory. So they said that this was a very nice way to test a theory in a very classical Popperian way of thinking, in terms of rejecting. It's because the model did a very precise prediction. We can do a very strong test in terms of falsification, right? So we can refute the theory if the slope is not -9, right, in this first approach. Of course, this was very simple, and they were very proud of the theory, but the problems started to appear. And a few examples and a few analyses start to show that the situation was not that good as was originally proposed in the Science paper. There was a different group. I was part of this group, as you can see here. So at that time, the things were much more complicated, but we were able to get together many data sets, and we actually showed that if we use different groups of organisms from different parts of Earth, we can have basically any pattern in terms of the relationship between temperature and species richness, right? So we have a prediction based on metabolic theory, but the slopes that we find in the real world were scattered around this prediction. So the problem is what we do. What does this mean? If we go back to the idea of Nancy Cartwright and all people that are proposing model-based science, this is not a big deal, but they are proposing this in a more Popperian falsification, strict falsification context. So we had lots of discussion in the literature about these results, and it's interesting because the Brown's group was thinking in terms of theory-based science, and we were thinking about model-based science. We were not aware of this kind of philosophical and epistemological idea behind the discussion. We thought that we're discussing the metabolic theory, but we are not discussing this. We are talking about a way of doing science. Because, for example, here in this paper, we found, let's say that we have less than 5% of the data sets that matched more or less the predictions of the theory, metabolic theory. So we said, okay, this theory is useless. And they said, oh, the theory is very interesting because it's able to predict 5%, a few of the data sets that are more well-behaved, right? So we were actually talking about a different way to do science and not about this particular theory or about latitudinal diversity gradients. And this leads, this discussion is just an example of this deeper philosophical problems underlying macroecology that now McGill pointed more clearly that we have two different perspectives in macroecology. We have that idea of macroecology as a broad-scale patterns that we want to understand. We want to understand the patterns, or we want to understand or to think that we have fundamental principles and natural laws underlying these patterns. So these are the two perspectives in terms of understanding macroecology. And now we can see that they are reflected in the way we do science. When we think in latitudinal diversity gradients in a more statistical and try to find variables that explain this pattern, we are thinking in a broad scale patterns that we want to understand them. But what Brown, for example, is doing here with metabolic theory is a search for fundamental principles, right? Is more similar to what we usually talk, for example, about physics, what the physicists are doing, right? So we have these two contrasting views of how to do science and how macroecology can be discussed, right? And this is very interesting, but it's not very clear that we are talking about different ways to do science. We are usually thinking about okay, we have these two goals for macroecology and so on, but there are more implications. What I want to say is there are deeper implications about this. And following this idea of these two ways of viewing science, we can think about the simulation models, right? So now we can go to the idea of, okay, we can think more deeply about representational models, and we can think that simulation models provide a very interesting way to think in terms of models as mediators between theory and the real world, because we don't have a theory. Actually, we have vague ideas about how the complex world works, right? And we also do not have very good data, because we actually do not have data or observed data. We have some representations of the real world. So models can be used to understand both, and our focus is the model. And simulation models provide a very interesting way, a very interesting example of this idea of models as mediators, right? And about the diversity gradients, the simulation models started with this paper by Rob Cowell and Dave Lees in the Trends in Ecology and Evolution. And Rob Cowell is a very famous, very important ecologist. He created, for example, the idea of new models in ecology. That is a very popular. I know that not all of you work with ecology, but those that work in ecology will recognize the term. New model is a term that was created by Rob Cowell. It's a more complex way to think about new hypotheses. And in 2000, he proposed what he called the mid-domain effect. In a first instance, it was a new model for diversity gradients. What Rob proposed is that the geographical ranges of species, for example, were not, the richness and the geographical ranges of species were not driven by environmental factors. They are just random. If the ranges are just randomly allocated in space, if you overlay these ranges to get the species richness, you have what he called the mid-domain effect, and that you can see here, for example, in a biogeographical domain in Brazilian Cerrado. We have here a mid-domain peak, okay? We have the highest richness in the middle of the geographical domain, and the richness decreases in a parabolic way towards the borders or the extremes of the biogeographical domain. And this is a simple consequence of the random overlay of the geographical ranges. So Cowell proposed this as a new model for understanding diversity gradients. So actually, rather than using a standard, let's say, a flat new expectations when we do, for example, a regression, we should use this parabolic pattern as a new expectation. And moreover, Rob Cowell proposed that not only that this was a new model, but this would be part of the explanation of diversity gradients. For example, if we think that we have more area in the tropics, species in the tropical regions would be more randomly allocated, so they are more constrained when we go to the temperate regions and when we go to extremely high latitudes. So he also proposed that part of the explanation for the high richness in the tropics was a random distribution of geographical ranges. And we had lots of discussions also with Rob Cowell and his group, and we have some exchanges in the literature. We did not agree with this kind of stuff, and in some way, mid-domain effect was not a lot of discussion. That was not very successful in terms of getting into the literature, and people did not actually accepted the idea of mid-domain effect as a new model or as a very important part of the explanation for diversity gradients. But when we started discussing this idea, it opens the possibility of simulating, because it's a very simple simulation that Rob proposed here to build the new expectation, right? Remember that we are talking about the year 2000 or before, so it was not very simple to do any of these things, for example, in a more computational basis, right? But what he proposes is that we have a randomization process of the geographical ranges of each species, and we should randomly reshuffle the geographical ranges of the species in the domain, and then we have this kind of figure that you have here, and this would be the expected richness under a random pattern in terms of diversity. And we can compare the observed pattern in species richness with this new expectation, right? And when he did this, he changed the way we use it to investigate the richness patterns, right? Because as I told you, what we usually did was to have the empirical pattern, we have richness pattern, and we have the explanatory variables, let's say temperature or productivity or range and elevation or some evolutionary measure. Okay, no problem. We can have this, and we did a statistical model to predict or to evaluate the richness pattern. It was a correlative model. But now, when Rob Cowell introduced the idea of mid-domain effect, what he was doing is to he was not using the variable, but it opens the idea that we can use an explanatory variable to model the range, and then we have the predicted richness that we can compare with the empirical patterns. So it's a minor change in terms of how we create the expectation, rather than correlating directly the explanatory variable with the empirical pattern. We use the explanatory variable. We started using the explanatory variable to create a simulation and then to generate the predicted richness. And this opened a very broad way to think in terms of the explanations, right? So Thiago Rangel, which he is now a professor here at the university, my colleague here, but at that time he was a undergraduate student, actually. He was a very, very good in computing, and at that time it was a very rare thing. And we started doing exactly this. Rather than correlating the temperature or the productivity with the richness, we started to use a more theory-based, some principles to understand how, for example, species would tolerate temperature or productivity gradients in terms of generating their distribution, and then we overlay the distribution under this kind of model, right? And then we correlate the model with the empirical patterns, right? So we have here, for example, we use the tolerance curve. Let's say it's a physiological basis for how each species is adapted to different environments, and then we can use this to generate the simulation models. And then different groups worldwide started to do this. We have the new expectation, but we can also use environmental variables to build range models to evaluate how species individually could respond to this variable, and then we can have the expected richness, right, in different scales and different approaches. And in a next step, we started to think about evolution. At that time, we just had environmental effects. So we are basically thinking in more static models, right? Each species would respond independently to the environmental factors, right? And then we have richness, but we did not have evolution in the system. And then by the middle 2000s, the new phylogenies and the new ideas on how species would respond to environment and speciate at that same time and extinct. And so we did the first simulation model in which we incorporated the evolutionary dynamics of the niche. So we are thinking now that species and niches are evolving in time, we are not simply talking about independent response of species, but the species are evolving under climate change. The climate is also changing, and we published this in 2007. Okay, it's another factor. Rather than thinking in static terms, we're thinking about speciation and extinction. No big deal. But in terms of the methods, we have a different perspective. So just for you to see how we model this, we have, for example, these two species here. We started with a single species, for example, this, let's say this red one. And what's happening is that we established those rules about how these species interact with the environment, let's say with temperature and productivity or precipitation. But, so we established the niche of the species. This is important. We have the bioclimatic envelopes, the limits that species tolerate. But then the climate changed. So we started here. We used a theoretical sinusoidal function. Sorry, a sinusoidal function on how. Sorry. A function of the environment fluctuating. For example, temperature increases, decreases, increases, and decreases. And when this happens, the ranges, or the species ranges, it starts to fragment, right? And so we decide what to do with each fragment. The fragment can go extinct, or the fragment can become a new species. We have an speciation processes. And then gradually we created diversity in terms of one species starts the range fragments, some of the fragments go extinct, and some of the fragments become new species. And when they become new species, we have to rebuild the species niche in terms of conserving the ancestral patterns or creating a new adaptation of the species to the new local environmental conditions. For example, this red species is very broad, but when we have this small species here, it can be in part, the new species can inherit the old niche, and in part the new species can adapt to the more local conditions. And of course, if we interact this during, say, hundreds or thousands of generations of species, we can have a simulated richness under this kind of processes, right? Okay, this is the basic idea. But of course, you can imagine that we have tens of parameters regulating all these processes. And so what we did here is one of the perspectives about how to use the simulation models to understand diversity. We looked for what are the combination of parameters that maximize, for example, the relationship between the expected, the observed or empirical richness pattern, for example, let's say with birds, South American birds. What is the combination of parameters that maximize the correlation between the simulated pattern with the empirical patterns? And we call it that, at that time, this strategy as pattern-oriented modeling. So we got a correlation of, say, almost 0.9, or right? That's the maximum, the best combination of parameters. Here we have just a few of the more interesting parameters. And then we interpret these parameters in terms of what we know about birds and what we know about South America. And we were able to, okay, it's a nice reconstruction of the observed. It's not actually observed, right? But the predicted model by under niche conservatism, speciation dynamics, and niche conservatism reconstructs quite well the observed patterns, although we still have a residual variation in the Andes. So in the Andean region, we have even more diversity. But this was very interesting because we used this pattern-oriented modeling, and we started to use the more complex evolutionary dynamics. And then mainly Thiago led this area and started to develop more and more sophisticated models, simulation models to do this. We also have this a parallel idea of neutral dynamics. So we are not talking about new dynamics, but we have the neutral dynamics in which we do not have adaptation to environment, but we just have dispersal and random extinction and random speciation. Is not exactly the new model, but we call this a neutral model. Most of you that work with ecology would be familiar with Stephen Hubble unified neutral theory of biodiversity and biogeography, which is actually a derivation of Kimura's neutral theory of evolution. It's a different interpretation in a more ecological sense. But this neutral macroecology idea, we can also use the same kind of idea, and we build an interpretation model. And we also show it, for example, a few years ago, that we have the same kind of pattern depending on the parameter combination. We can also lead, the combination of parameters also lead to a pattern similar to what Cowell call it mid-domain effect, right? And it's interesting, and more recently, one of our PhD students, Marco Tullio, showed that this neutral expectation leads to a pattern in South America that also reconstructs well the observed or empirical richness patterns. But as we can see here in the circle, now we are starting to adopt a slightly different perspective in terms of how we use the simulation models. Okay, we can go and we can reconstruct and try to reconstruct empirical patterns. We can try to see what is the combination of neutral dynamics and other environmental parameters. We can combine both ideas to generate the prediction. So we have more theorizing process. We generate this representation model, and we can see if we match the observed or the empirical pattern. But here, what Marco Tullio is showing is that random dynamics can, a neutral dynamics can generate a phylogenetic signal in the data that is usually interpreted in terms of niche conservatism. The niche is conserved, is evolving very slowly, and then this creates a diversity pattern. But here we are using the model to show that even under a purely neutral dynamics, we can also find a phylogenetic signal in the niches of the species due to geographical constraints. So here we have a slightly different way to think about the models. We are using the models to show that the empirical interpretation of the phylogenetic signal in the niches, in the ecological niches of the species, is not, maybe not necessarily what we are thinking, right? So this is a slightly different problem in philosophy of science that is called underdetermination. We can have different theories that leads to the same empirical patterns. And and more recently also Thiago, that you see Thiago here, and Rob Cowell, now we are friends, now after we fighting about the mid-domain effect. Soon Thiago went to US to study with Cowell, and so we were able to join the ideas of mid-domain effect, and this started all this thing that I showed you about using the simulation of ranges to generate richness patterns. And we published this paper in Science a few years ago in 2018. And in this final model, final in terms of what we were improving those models that I showed you. We are incorporating more and more drivers. Now we have biotic interactions. We have the real, this is important, we have the real dynamics of the climate, real in terms of the paleoclimatic models. We have some of the authors were paleoclimatologists, and they reconstructed the climate changes in South America in the last 5 million years, right? We use it 1 million year here, but we have 5 million years now. And this is more realistic as a model of data that is being used as input. And we have all there are a lot of other factors. For example, species are explicitly adapting. It's an eco-evolutionary dynamic model. Species are adapting to each environmental conditions in different regions of the continent. And this is finally a more model. The idea here in this paper, although we did some comparisons, mostly because the reviewers asked for this, we did some comparison with bird richness or tree richness or mammal richness, but the main focus of the model was to study patterns, abstract patterns of diversity in South America by shifting and by changing the processes that were activated or not activated in the simulation. And for example, here you can see one example of one animation of the evolutionary dynamics. And here we have the niches evolving in this figure here in terms of temperature and precipitation. And we also have the phylogeny of these species here. And we can use all this information in terms of the richness and the phylogenetic structure to see how they match the real data. And more important, we can understand how different processes that were incorporated in these very complex models affect the possibility of observing in different clades, in different groups of organisms, different patterns. For example, if we have a clade that started, the first species started in the Andean region, we have one expected pattern in the end, and we also have different patterns if we use the phylogeny to deconstruct older species or new species, and then we calculated diversification rates or regions in which we have more extinction because the species are not well adapted to those regions. So we have Andean founders, Atlantic forest founders, Amazonian founders. So we used the models to study the possibilities of finding different patterns in South America. Also, another thing, for example, Thiago simulated erasing or flattening the Andean region. If we do not have the Andean region, this is some kind of counterfactual idea. If you do not have the Andes, how would be the pattern under different combinations of parameters? So I think that's very interesting in terms of shifting the perspective of using simulations not to describe the real world or not to represent, or not only to represent the real world, but also as a way to study possibilities and to study potential of how diversity would appear in this particular region, right? And so, of course, this is also an ongoing, ever-going idea or process. We have two very different new papers that appear this year, last year, using more and more sophisticated platforms. For example, Genesis is proposing to have a platform so everybody can do this and everybody can try to do its own experiments and see and evaluate diversity patterns. They're trying to do this kind of thing. And another very interesting interface is how to couple real local adaptive processes and broad scale deep time dynamics. So this is still very challenging. And the other line that I'm working with in the last few years is a more genetic adaptation, in terms of genetic adaptation, phenotypic plasticity at population level, and how this can be used in terms of simulating macroecological patterns. I've been studying, for example, body size evolution in islands. This kind of thing is more phenotypic, local scale process. And one of the challenges is just is exactly how to combine this local process with broad-scale dynamics. So we are actually now working on a new simulation model that tries to incorporate more and more combination of local and broad-scale, local adaptation, local evolutionary process with broad-scale speciation and extinction dynamics. Okay, that's what I was planning to talk to you today, and of course I'm open to questions, but I think that this model-based reasoning that we are pursuing, I think it's a very interesting perspective to work with macroecology. And I think that we can apply this kind of reasoning to understand what happened in this field of diversity gradients in the last 50 years. And we can now have a more critical, and we can understand the debates that happen if we understand these different ways and different perspectives in terms of doing science, and more specifically in terms of how to use the simulation models to understand both the theory and the real patterns or the empirical patterns that we see in the world today. Okay, thank you very much. I'm open to questions if you understand, if you have a minimum understanding of my terrible English. Sorry for that.
Thank you, José Alexandre. I think, do you have questions? Yeah, get the microphone. Where is the mic? One, yeah. Say your name and then make your question, okay?
Yes. Well, my name is Ruth, and well, my question is related in what type of models do you use to study the richness? I mean that well, I see that diffusion in the land. So I was thinking about differential equation, partial differential equations, or maybe you use models related to machine learning. So, well, my question is about that.
Oh, I see. What we're doing is to establish some rules. We are not using differential equations. We are using, how can I say this? We are just defining rules in terms, for example, all these models are defined in terms of, we started with the ranges of the species, right? It's very simple, right? We say, for example, that the species is adapted, the optimum of the species is at 25 degrees of temperature, and we have a standard deviation of 2, right? So we randomly choose one cell within this range, and we use it a spreading die, a cellular automata. It's a cellular automata, right? We grow the range of the species within these limits. And then we repeat this for different species, and we have different rules, for example, for the optimum. And then this is a very simple way to create one richness pattern. But I think that, if I understood your question, the basic idea is that you use cellular automata models, right, with different rules to occupy the space and to propagate in time, right? We can start with a single species, and then the environmental change. If the environment changes, the range fragments, and we have to decide what to do with of the range fragments. That's the idea, right?
Okay. Yeah. Can I?
Yeah.
And how did you combine, well, temperature and waterfalls and well, all others parameters that you mentioned in the models? How did you add that kind of characteristics in the parameters?
We have this in the multidimensional space. We have defined centroids and we are not using a variance-covariance matrix. We are thinking in each one independent. This is a challenge because we do not have empirical covariances between among the variables, right? So we have independent rules for each of the explanatory variables.
Okay. Thank you.
More questions? Yeah, the mic. Here.
Well, hi, Lola. In the beginning, you talked about there being a distinction between theory and model, and I would like for you to comment a bit on that and expand it, because I couldn't really grasp what you meant by it.
Okay, so, sorry. This is a complicated issue, and I'm just starting in terms of. I think this is the first time I talk about this in public, and I think that the idea is that we have a lot of confusion in the literature between what we call theory and what we call model, right? This is a classical problem if you go to the literature in philosophy of science. But perhaps the idea of theory, if you go to that paper that I showed you in Oikos by Paulo Inácio, Flavio knows him very well, Paulo Inácio and Renata and the people from São Paulo and from Bahia, you can have a better idea. But the idea of theory is that we have an axiomatic approach, and the models are an interpretation of the theory. For example, if you go to the metabolic theory, to James Brown's metabolic theory, they have general principles, and they start to creating. There are models inside. There are natural laws. There are concepts, and you are axiomatically putting all this together. Then you derive and you go to a model, an equation, right? So we have a prediction that under that theory, if everything is working with the theory, there will be a negative relationship, an inverse relationship between richness and temperature with a particular slope, right? So sometimes people call this an interpretative model, interpretive model, because it's a model that is derived from a theory, right? But the criticism about the theory is that when you do this, you are not necessarily focusing on the real world. You are modeling the theory, right? And you expect that your ideas about the theory are correct, and then you go to match the real world. That's a very standard way to think about science. But if you think, well, this is not what we are doing, at least it's not what we are doing in macroecology and perhaps in ecology in general, as Paulo Inácio said. What we're doing is to try to understand the world. We are not trying to understand the theory, right? So there's a subtle inversion in terms of the direction from where model comes on. And then I found out that people were discussing this in physics, for example. They are not talking about the physical theories. They are talking about how to represent the world. So it's a different way to think in terms of the model. So models gain more independence in respect to the axiomatic idea of a theory. So, for example, Ronald Geer do not talk about the theory. He talks about the theorizing process in terms of, okay, we have some ideas, we have some concepts, some ideas, some principles, some limiting conditions, and then we can use this, and we can use, for example, ideas from different fields. Here, for example, in the science paper, we had many things that came from quantitative genetics. Something came from climatology. Some came from, macroecology, range dynamics, speciation, evolution. But we do not have an axiomatic theory that we derived to construct the model. So that's why I tried to show you that simulations are a very nice example of this independence of theory from the models, right? Theories and models. So models become the focus of the research, right? And theories is just a way to build a model, right? So that's why people is talking about, let's say, theory-based reasoning or theory-based science versus model-based science, and or a model-based reasoning in terms of course, when you do a model, for example, a simulation model, and we contrast this with the empirical patterns, of course that we gain theoretical understanding, right? But this is not the focus, right? This is not the main goal. The main goal is to study the real world indirectly by the simulations, right? Okay. I think that's the idea.
It's clear now, but I'll check the paper.
Okay, fantastic.
There's another question.
Hi, my question is more specific about something that you said in your lecture. It's something that stuck in my mind, and I don't know if I missed something. But you said that when talking about neutral dynamics, I think in evolution, that random dynamics can generate a phylogenetic signal. And I was wondering, how do we interpret that, the phylogenetic signal, and what could be the causes of that, because it's random, you know. And
Okay. That's a great question. I was very brief about this, so that's great that you asked. The issue is that many people is measuring phylogenetic signal in niche dimensions, for example, temperature or any multidimensional metrics that you want to do in terms of species niche, Grinnellian niches, in terms of the environment and so on. And when we find a phylogenetic signal in niche, and when we map this in geographical space, and we find this, we think that the diversity gradient is driven by niche conservatism, right? What I would say, for example, the classical idea that was proposed in 2004 by John Wiens and Donaghy is that, for example, tropics have more species because ancestral niches were tropical, so the niches tend to be conserved, so species accumulated in the tropics, and eventually some of these species are able to adapt to temperate regions and to diversify there, but we have an accumulation because niches tend to be conserved, and this appears as a phylogenetic signal, right? So what Marco Tullio showed that in that paper is that because of the geographical constraints and the spatial structure in the environment, niche conservatism will appear anyway, even if richness patterns is driven by neutral dynamics and not by, there's nothing to do with niche. The niches are evolving randomly, but the geographical structure in the environment creates coupled with the geographical component in the speciation, right? Because these species are not appearing anywhere. These species are appearing geographically close to the ancestral species. We have some kind of collinearity effect generated by space, right? Because the environment is structured in space, it will create it creates a phylogenetic signal anyway, even if the dynamics is not driven as in our other models by niche adaptations, by niche evolution under climate change, right? Okay, did you get it?
yes, thank you.
There's another question.
Hello, my name is Thales. Well, first I would like to thank you for the lecture. It was really nice.
Thank you.
I have like two connected questions. First, these models that you are using, they are complex in the sense that you have many things, but they are not as nearly as complex as the real life is, and models are not supposed to be like this. But
Sure.
what do you think we can, how this, because these models have a more or less, by the maps you showed, a good accuracy of like representing the patterns that we are seeing. You have some local things that are not matching, but in the general pattern is really amazing that you can generate this by a model that is running with all these parameters. And what this make you think about this process that you can generate this species distribution and richness patterns using these simplified models that that are simple in relation to the nature, and in relation more about the models. What do you think are the next things that we should implement, or that you are working now in this model? Some important processes that are still missing.
Okay, that's great. Let's go. Well, about your first question opens a very interesting discussion. I'm also quite interested. I briefly talked about this too. Is about underdetermination, right? And about the realism underlying theoretical ideas and how this goes to a model. The problem that we have here, you told, for example, that we have some parameters. It's not that complicated, but we are not talking about 2 or 3 parameters, right? We have lots of parameters, depending on how we define them. But okay, it's not very complicated. But the problem is that what about different combinations of parameters that could generate very similar patterns, right? So this is the underdetermination issue, and this leads to the idea of how realistic we can think about the match between the empirical patterns and the simulated results. We tend, in general, as ecologists and natural sciences, I think, we tend to be realists, right? In a philosophical sense. If we find this match, we think that we found something. That there's something there that must be real, right? Because the problem is not the empirical or observant pattern. The problem is the underlying unobservable patterns. For example, in our case, we have evolution, we have speciation events, and we have extinction events. And of course, our simulation is not reconstructing particular events of speciation. We are generating the speciation dynamics, but not the speciation or extinction events. And this leads to the same pattern. And of course, although we know that we do not have exactly the same events, we know that the speciation dynamics seems to be important, right? So if we have this match and we, for example, generate a flat pattern of speciation and extinction, even if we have speciation and extinction, these processes should not necessarily be spatially structured. Would this generate the same pattern? No. So we think that we have something, but of course it's not the same. So this is a form of underdetermination, and so it's a warning about how confident we can be about discovering, for example, fundamental laws, right? So that's a very interesting discussion in terms of how confident we must be in terms of finding real process, unobserved process, and how they explain explain or they really happened in the past. So this is a very interesting philosophical problems that I think that also simulation models can help us to deal with, right? It opens many possibilities of discussion about this philosophical issue of underdetermination. That is, it's very complicated to think about this, especially in broad scale and non-experimental historical parts of the reality. And in this sense, for example, Ronald Geer's ideas that what he called perspectivism, right? So if macroecology is a perspective, a way to see the world, as you said, although we have relatively simple process parameters, we are not seeing exactly the same speciation and extinction events. We are able to reconstruct the world. So this is probably part of the reality. We are looking for one perspective that is able to help us understanding the part. And understanding here is a psychological, emotional perception, right? I'm satisfied with this, right? I think I understand this and I'm happy, right? So we are talking about happiness actually. And this leads to your other questions, or the second question, what we are doing now in this line, and following this idea of realism, actually, for example, what we are discussing in terms of improving the models, right? So if we're improving the models, we are thinking about finding more realistic parameters and more realistic processes. We are thinking that we can, rather than modeling the ranges of the species, because now we have the ranges of the species, right? We have the ranges as a unit in terms of the simulation, right? Okay, it can go fragmented, and when it goes fragmented, and we have a parameter to say that, okay, this is a fragment of the range, and then we decide to do what with that particular fragment. Now, what we want to do is to simulate populations, right? To go to a lower level, a lower hierarchical level, and go to populations. And then we want to see how the properties of the geographic ranges of the species will emerge from the process at a lower hierarchical level. I think this is a challenge at this scale, right? At this scale in terms of geography and time, right? We can, of course, we can simulate the populations, and our group also have several papers and analysis at geographical level, working with population genetics, for example. Okay, no big deal. But how we can combine this dynamics within the ranges, and how this happening for many species at the same time in very deep time, how this will help us to reconstruct or improve the reconstruction of the macroecological pattern. So we are now thinking about this, let's say, interscale models, or combining models in different scales to see how macroecological patterns emerge. Right? Hopefully.
Can you listen?
Okay,
yeah?
now.
Sorry. Is there any other question? On the back. Can you pass the mic?
Hi. Can you hear me?
Hi,
my name is João. Again, thanks for the presentation. It was really, really good. I wrote down a few questions. At first, when you were talking about defining a species range for these cellular automata models, you're talking about the range in geographic space, right? And then just translating it. Yeah, that was the first question. And I got really interested in the science paper with the simulations. And I was wondering if if we can get those models, like if we could get those layers and those simulations, or at least the code to reproduce it. Is that data open?
I don't remember if the code is really open in it's Thiago programs in Delphi. It's very odd, I think, but I think you can talk to him. No big deal. There's no secret behind it. But it's not. I think this may be a problem. I think this is not open code in R or in Python or this more popular languages, because Thiago writes in Delphi. So I think it's Pascal, right? I remember right? But we can talk to him, no big deal. But for example, that other platform, they are working with the group from Switzerland and Germany, the Genesis. It's I think it's a more open language and open code stuff, and you can do different combinations of things. And it's more friendly user than what Thiago is doing, right?
Okay, thanks. More questions?
My name is Amanda, and could you talk a little bit of the pattern-oriented model and if it could work with microevolutionary process?
Sure. I really like the pattern-oriented modeling, and I'm actually a bit proud that we thought that we invented that. It was very naive at that time in 2006 or 2005. We had this idea of we were very amazed because we had this simulation system, and I thought to Thiago, okay, what about generating random combinations of parameters, hundreds or thousands of parameter combinations, and trying to find which one gets to the better match with the empirical pattern, and then we can interpret which parameters are doing, and when we did this, it did a lot of sense, right? In terms of what we were thinking about niche conservatism and so on. Oh, that's fantastic. Then we submitted that to American Naturalist, and the editor was Volker Grimm, that worked with individual-based models and behavioral ecology and all the stuff. And then he told us, okay, that's great, but this is what I call it pattern-oriented modeling, right? But we are doing this at macroecological scale, and he liked it. But for example, he insisted with us to use his own protocol to describe the model steps and to adapt what they did to individual-based models, agent-based models in a more general sense, but we usually talked about individual-based models in ecology, to our macroecological analysis, right? But the idea was that, right? We generate random combination of parameters. We did this lot of things, and we try to see which combination of parameters goes to a given target, right? So this was the idea of pattern-oriented modeling, and of course we are not talking about, let's say, a single pattern, because if you go to a single pattern, it's a single dimension optimization, right? For example, I just showed, for example, the best correlation with richness pattern, but we can also look at the same time if the species ranges frequency distribution also matches, or if the phylogenetic structure also matches, so we can look for multiple targets in pattern-oriented modeling. And for example, I still use, because I know this and I think it's fair. For example, I just used this in this other studies using simulations for island rule, body size evolution in islands that I briefly talked to you about. It's a microevolutionary model with adaptation and population changes, and the phenotype is changing. The body size of the species of the population is going small to when the species occupy an island. So it's a microevolutionary model. We just published this in Journal of Biogeography. And I used the pattern-oriented modeling in terms of that we had we know that we simulated the evolution of a cervus, a reindeer, red deer in the Channel Islands, that we had a nice fossil record, and we knew what was the time window of the island isolation that triggered the dwarfing of the body size. So I used the pattern-oriented modeling to find which combination of population and genetic and demographic parameters would drive body size evolution within that time window, right? So I used the pattern-oriented modeling. But for example, now Marco Tullio is doing this using MCMC, Monte Carlo chain Markov algorithms, and not a random search. So he's using MCMC to look for which combination of parameters maximize in terms of the optimization, or to describe the distribution, the a posteriori distribution of each parameter, right? So it's basically the same kind of algorithm. We can use more sophisticated and powerful algorithms for achieving the same goal.
Thank you.
There is one question on the other side. Sorry.
Hello. Okay.
Hi.
Hello. Okay.
Okay.
Hi, my name is Cristiana, and you were talking about the type of models that you use. That you say were like more simple in a mathematical sense, but do you have any plans of working with a more complicated or in like statistical models that you are interested in working to kind of deal with these scenarios predictions that you were showing us?
Sorry, it was cutting a bit. I'm not sure I understood. Can you repeat, please?
Okay. And the microphone doesn't like me. So you were answering before the root questions that the models that you use, the ideas, are like simple. But do you have any other models that you would like to use, or any statistical modeling that you are interested into, that you want to work in these scenarios predictions that you show us?
Oh, I see. I think I understood what you mean. I just talked to you about the simulation stuff, right? But in a more, let's say, daily basis, what we usually did was to do a very standard statistical approach. We have species richness, and we have, let's say, temperature, we have productivity, we have, now we can use the phylogenies of the, let's say, birds. We can have the phylogenies to estimate speciation rates for each lineage, and we can map these speciations, and then we can do correlative models, right? This is a completely, it's not very different if you go to the Ronald Gear's perspective, but it's not a simulation, it's empirical, everything is empirical. And then we have the explanatory variables that they came from the theorizing process. We can think about, let's say, physiology, and we know how, let's say, amphibians work. Amphibians are different from reptiles, that are different from birds in terms of temperature tolerance or productivity or precipitation. They have different dynamics because their phylogenies have different shapes. And if we think about everything, all this kind of ideas, we can generate surrogate variables, empirical surrogate variables that we can use to
Is it working? No, you can say. Yeah.
Okay, so about tomorrow. So tomorrow we don't have journal club because we thought that I told you we were going to have one in my class. We're going to have it on Monday. I was looking wrong to the program, but it's fine. But basically what we're going to do in the afternoon is from 2:30 to 4. We're going to have like a Q&A session, discussion session, so you can ask me things about the course. I will be here. And then after that, at 4, Guy is going to help you also with more computational problems that you may have with the master equation, especially because she speaks your language computationally, which is Python and things more common than all the stuff that I do. Those 2 sessions, 2:30 to 4 and 4, let's say 1.5 hours, 2 hours for on, those are, how do you say, volunteer. Okay, so those of you that when I open the master equation, you feel that you already, it's very slow and it's boring, or you already learned it, or you don't have to be here. Okay, if you have doubts about things that you've been studying further beyond the course, you can be here and you can ask, or you can ask me other day. You don't have to be here at 2:30, but if you have questions about the course and want to ask them, I will be here and we can manage all these things together. From class 1 to today, more or less, which is more or less the same, different systems, okay? And there is Monday as well, or? On Monday we talk about Monday. Okay, perfect. So that's it for tomorrow, okay? No journal club, no need to read. You've been reading a lot.
Just a second. José Alexandre is trying to get in by his cell phone because then he uses the battery and the cell phone that is providing the internet. But I don't know if it's working. Just give me 2 minutes. Just to not make like a big scene here. Hi, José Alexandre. Testing. 1, 2. No, maybe.
Oi.
Yeah.
Agora acho que deu.
Do you listen? Yeah.
Okay.
Do you want the student repeats the question again?
Let me remember. No, okay. I was talking about the surrogates, right? So we have all the surrogate variables, right? And then we go to very standard statistical analysis, right? So then we have a different way to think about the models because we have an empirical statistical model, and then we go to the statistics. So, for example, now we are using a lot of structural equation modeling. In the past, we just used it, for example, multiple regressions to evaluate, and all the ideas about using archaic information criterion to select which is the best model, the best explanatory model. but now we have all these ideas about using structural equation modeling, and we also worked a lot with the spatial statistics, because there's a lot of confusion in terms of statistics because we have the spatially structured data, right? So it's a different world, right? And then of course we are doing this for particular groups of organisms. Is this working?
José Alexandre, it was my fault. It's working. It's just the image that is in loop. I don't know how I did this. I'm
It's
so
great.
sorry. But you can answer. They're listening you.
Oh, okay. Did you listen to my answer about the statistics? Okay, or no?
Yes. Yes.
Yeah. Okay.
We have time for one last question, if anyone wants to make it. No.
Oh, now it's more confusing.
Sorry, now they see you. I have a quick question. Can I make it?
Sure.
Am I allowed, guys?
Sure.
So you talked about going from a lower scale to a greater scale in ecology. And I've been working evolutionary models, and it seems it's a gap everywhere, right? Going from micro to macro, micro to macro. It's always a problem. And do you think the same processes that are happening in ecology are also happening in evolution to make this going from one level to the other level? Do you think they are similar?
No, I think that's part of the problem, right? Because, for example, if we are thinking about first the diversity gradients in South America, this is completely different about talking the difference between richness between, let's say, one region in Atlantic forest to South Atlantic forest and North Atlantic forest. It's completely different in terms of the process. Or if we think about local, because actually we are talking about diversity in a large grain. But if we go, for example, to a local sample, we must think about biotic interactions, because, for example, in this science paper, biotic interaction really doesn't matter. It does not change anything if you do not account for biotic interactions. But if you go to local scales, you must think about biotic interaction. You might think about habitat heterogeneity, right? Local beta diversity. So I think that part of the problem is that we have different processes, and the impact of these processes is not the same across the scale. So this is a more conventional strategy. And from the philosophical point of view, I think that's why it's interesting to think in perspectives, as Ronald Gear said. So macroecology can be viewed as one perspective. So it's difficult to combine this with a different, let's say, more local perspective, right? So I think we have limits to translate one model from one level to another level. What Tiago and I have been discussing about this, and Tiago is quite interested about this scale shifts in terms of modeling. Our final discussion, at least as our point of view now, is that although the models are not necessarily the same in terms of the parameters and what is the variables that are important, I think that they must be coherent, right? They have to be, for example, within the same framework and represent more or less the same kind of and be built in the same kind of with the same kind of strategy. It's a question of coherence of models between the two levels, but I'm not sure that we can have one single model, for example, for everything. I'm not sure about this. Both in terms of some operational stuff. For example, we can. It's very difficult to think in an individual-based model for macroevolution, right? I'm not sure. For example, in our quantitative genetics studies, it's very common now, people simulating microevolution in terms of quantitative genetics, using, for example, Mendelian segregation of hundreds or thousands of alleles and simulating the dynamics of the alleles in the population. But it's not necessary because we have quantitative genetics, but just have to work with means and variance, right? So I think that we must understand one model in one level with what is happening in terms of coherence. We can understand the model in one level by the processes that are more likely happening in a lower level, but we not need to model these local processes, right? For example, if we're thinking about, let's say, body size evolution, we can do this using means and variance and covariance and irritability and so on. We know that the basis is Mendelian, but we do not need to go to a Mendelian simulation to get body size evolution. That's not really necessary. It will add almost nothing, at least from our perspective, our macroecological perspective, right? I don't know if I answer. It's a bit confusing anyway.
No. It's great. I think it makes a lot of sense. And when I was thinking ecology and evolution, so there is something that is like the time or the time scale that makes the difference between ecology and evolution. So I was thinking maybe the processes in ecology, micro to macro, and the processes in evolution, micro to macro, are about the same, but there's something that links them that is the time scale. So I think I'm thinking a lot.
Okay, okay, I see. There's another issue here. There are all the macroecological processes and contingencies and emergent properties. Okay, there's more complications here. I agree with you. I see your point, right? This is very challenging, I think, how to look for these emergent properties in terms. For example, I think that we have something here. I was talking to Thiago a few days ago about the speciation dynamics, right? We are simulating the speciation dynamics in a very naive way. And one of the goals that I mentioned about moving to a lower level, at population level at least, is that to have more realistic and more possibilities of simulating speciation in a more complex way in terms of leading to evolution of niches or leading to evolution of traits, changes in traits, in species traits, in a more sophisticated ways, because we are not doing a good job in speciation. And I think in part is what you were talking about in terms of time scaling and about having more complex emergent properties at species level than what we have at population level.
Yes, yeah. Yeah, I think you got it. And I understand it's complicated. But I'd like to thank you for everything so far. We hope to have you here again in the future. And try me to thank him.
Okay, I'm not listening quite well. There's something.
They're clapping now.
Okay.
Okay, thank you, José Alexandre.
Okay, bye-bye, everyone.
Bye-bye.